Hi everyone, and welcome back to my A-level biology lecture series. In the previous lesson, we have seen the overview of the meiosis process. So, in this lesson, we will continue to explore the processes or the phases during meiosis 1. Let us start with the interphase, which is the preparatory phase before the meiosis process. Interphase occur before meiosis begin, and it is divided into three subphases, which are G1, S, and G2. It is similar to or same as the process that happened before the mitosis. During G1, also known as the first gap phase, the cells grows and synthesize protein needed for DNA replication, such as helicase, DNA polymerase, ligases, and so on. During S phase, which is also known as the synthesis phase of interphase, DNA replication occurs by a semi-conservative mechanism. This means that each new DNA molecule consists of one parental strand and also one newly synthesized strand. This we have learned in DNA replication previously. As a result, each chromosome is duplicated to form two genetically identical sister chromatids, which remain joined at a central region known as centromere. During the G2 phase, or known as the second gap phase of interphase, the cells undergo final preparation for meiotic division. The cell synthesized protein required for chromosome condensation as well as spindle formation. The cells also check for any DNA damage before entering meiosis so that the damaged DNA, mutated DNA, wouldn't be passed on to the gamete. And this is very dangerous because once this damaged or mutated DNA is passed on to the gamete, it will affect the next generation. In animal cells, the centrosome is duplicated during the G2 phase of interphase. And each centrosome consists of a pair of centrioles, which help to organize the spindle microtubule to form spindle. So these spindles are essential for the accurate separation of homologous chromosome during meiosis 1. And the diagram actually shows a cells or an animal cells at interphase. So how we know this is interphase? And we can see that during interphase, the nuclear envelope remain intact, the nucleolus remain intact, and the chromatin has not or have not condensed into chromosome yet. Okay, so this is a G2 because we know that the centriole or centrosome, okay, duplications of centriole and centrosome occurs and completed at G2 phase. Next, let us look at meiosis 1 with the function to halve the number of chromosome, and that's why meiosis 1 also known as the reduction division. Meiosis 1 is the first division in meiosis. It is known as a reduction division because it reduces the diploid 2n chromosome number to haploid after the first meiotic division. It consists of four main stages, which are prophase 1, metaphase 1, anaphase 1, as well as telophase 1. The role of meiosis 1 is to separate homologous chromosome, therefore reducing the chromosome number from diploid 2n to haploid n. And during the process of meiosis 1, genetic variations is introduced through these two main processes, They are crossing over that happens during prophase 1 and independent assortments of homologous chromosome during metaphase 1. So what are the key features of meiosis 1? One of the key features is during prophase 1, homologous chromosome pair up to form bivalent and undergo crossing over at point known as chiasmata. This event introduces genetic variation. And this is a key features of meiosis 1, where we won't be able to see this during mitosis, prophase during mitosis, as well as the prophase 2 of meiosis 2. And in anaphase 1, these homologous pair are separated and pulled to the opposite pole by spindle microtubule. Compared to the mitosis, anaphase in mitosis separate sister chromatid. And during anaphase 2 of meiosis 2, sister chromatids are separated. So this event, homologous pair are separated, is a key features of meiosis 1. It is special for meiosis 1 only because it happened during anaphase 1. So after telophase 1 and cytokinesis, the cell divides to form two haploid daughter cells. And each haploid cells contain half the original chromosome number, but each chromosome still consists of two sister chromatids joined at centromere. And because of this, the cells need to proceed to meiosis 2, where sister chromatids are separated. Let us look at a prophase 1. So the cells in prophase 1 shows features of condensations of chromatins to form chromosome. The nuclear envelope disintegrated, and you can see that this a pair of centriole move to the opposite pole, and when they move to the opposite pole, they organize the spindle microtubule to form spindle. And due to the condensation of chromatin, nucleolus disappear. So if we get the human cells as example, so the number of DNA molecule per human cell is 92. This is due to DNA replication during S phase because each 46 chromosome consists of two sister chromatids after DNA replication. So what is the number of chromosome per human cells? Yep, 46 chromosome, and these will remain unchanged during prophase 1. So what are the behavior of the chromosome? First of all, chromatins condense and become shorter and thicker, forming distinct metaphase chromosome, which consists of a pair of sister chromatids joined at the centromere. And metaphase chromosome is visible under a light microscope. So each metaphase chromosome consists of two identical sister chromatids joined at the centromere. And homologous chromosome now pair up to form bivalent or tetrads. Bivalent because two chromosomes pair up, and tetrad because of four sister chromatids. So after this homologous chromosome pair up to form bivalent, okay, crossing over occur between non-sister chromatids of homologous chromosome at a point known as chiasmata, and this event, crossing over, introduce genetic variation. So kinetochore form at the centromere of each chromatid, and this kinetochore is very important because it provides attachment site for spindle microtubule. In the late prophase 1, spindle microtubule begin to attach to the kinetochore, positioning bivalents or these tetrads for alignments on the metaphase plate in the next stage. So we have to understand that this kinetochore allows the attachment of the spindle microtubule, and the elongation or the formation of spindle microtubule helps to push these homologous pair and make sure that they align at the metaphase plate during metaphase 1. How about the centrosome? Please bear in mind that centrosome only appear in animal cells. In plant cells, please do not include centrosome. So the pair of centrosomes start to separate in the beginning of the prophase 1 and migrate to opposite pole of the nucleus. Spindle microtubule begin to form from the centrosome, preparing for the chromosome movement. Nucleus disappear as the cell progress through prophase 1. Nuclear envelope begin to disintegrate into a small tiny vesicle, and this allows spindle microtubule to access the metaphase chromosome. And this microtubule attached to the kinetochore located at the centromere of the metaphase chromosome. And as I mentioned previously, the elongations of the spindle microtubule push this homologous chromosome to the metaphase plate. And plasma membrane remain intact during prophase 1, and no special event is associated with the change in the plasma membrane. There is no change in the plasma membrane throughout the prophase 1. This is extended learning where we look at synapses, where the homologous chromosome pair up and then crossing over, and the formation of chiasmata. So in the early prophase 1, we know that homologous chromosome begin to pair up. And each chromosome seek out its homologous partner, the one with the same gene in the same loci, but possibly with different type of allele. And we have this complex known as the synaptonymal complex formed between the homologues, physically align them side by side. This very important protein or complex allows the homologous chromosome to pair up. So together, the homologous chromosome now form a bivalence. As I say that, bivalence means 2 units of chromosome, which they are homologous. And we use the word tetrad because consists of 4 sister chromatids. So after they have paired up, the next event is crossing over. So as synapses completed, homologous chromosomes are closely aligned, and this allowed non-sister chromatid to exchange genetic material. So at a specific point, matching region of non-sister chromatic breaks and rejoin, resulting in crossing over. So we can see that in the diagram. Okay, so step number 2, where crossing over take place. So the blue color DNA and the red color DNA now, they exchange the genetic material, or more precisely, actually, they exchange the allele. So enzyme, for example, like endonucleases cut the chromatids, while ligases help to seal the exchange segment. Endonucleases are the enzyme that cut or cleave or hydrolyze the phosphodiester bond, and ligases are the enzyme that form or seal the sugar phosphate backbone. So through this crossing over, the crossing over produces new allelic combination. So where a single sister chromatid now contains paternal and maternal allele within a sister chromatid, and this increases the genetic variation among the gametes. So as prophase 1 progresses, the synaptonymal complexes break down and homologous chromosomes begin to separate slightly. However, at the point where crossing over occurs, the chromatids remain physically linked. So these egg-shaped connections are known as chiasmata, and singular is known as chiasma. So the chiasma ensure that homologous chromosomes remain connected until they are separated later in meiosis, which is anaphase 1. So the diagram shows that the formation of the chiasmata, which join two homologous chromosomes together during prophase 1. Now, let us look at what are the events that take place during metaphase 1. So during metaphase 1, the signature events that take place, we can see that the homologous chromosome now pair up at the metaphase plate. And due to the movement of the centrosome or centriole, so the spindle microtubule is organized to form a complete spindle. We won't be able to see any nucleolus as well as the nuclear envelope, because the nuclear envelope is completely disintegrated and nucleolus is completely disappeared. So what is the number of DNA molecule per human cells? So in every single human cells, in this case, it is 92. Why? Because again, each chromosome or metaphase chromosome consists of 2 sister chromatids, and each sister chromatid consists of 1 DNA molecule. And there are 46 chromosomes per human cells at this moment, and this number will remain unchanged throughout the metaphase 1. So what are the behavior of chromosome? So first of all, homologous chromosome pair are aligned along the metaphase plate, and metaphase plate also known as the equator of the spindle. And each homologue in a pair is oriented toward opposite spindle pole, and this arrangement allows for independent assortment, contributing to genetic variation. So at this moment, do not worry about independent assortment. At the end of this topic, on the next topic, we are going to learn about how independent assortment as well as crossing over that helps to contribute to genetic variation in the gamete. For each metaphase chromosome, kinetochore on sister chromatids are attached to spindle microtubule from opposite pole. And this ensures that homologous chromosome, but not sister chromatids, will be separated during anaphase 1. So each chromosome will get 1 spindle microtubule attached to their kinetochore, unlike during metaphase. In mitosis, each chromosome will receive 2 spindle microtubule that attach to 2 kinetochore on the sister chromatids. Centrosome in animal cells only, and the centrosome are now positioned at opposite pole of the spindle, and each centrosome organize and extend spindle microtubule towards the metaphase plate, where they attach to the spindle microtubule attached to the kinetochore. So spindle microtubules are fully formed and extend from each centrosome, and they are attached to the kinetochore of each homologous chromosome in the bivalent. Nucleolus is completely disappeared at this stage. Nuclear envelope is completely disintegrated, and there's no event associated with the plasma membrane, and plasma membrane remains intact during metaphase 1. How about anaphase 1? So during anaphase 1, you can see that, okay, the homologous chromosome are separated because the breakage that happened during or at the chiasmata. So due to the shortening of the spindle microtubule and spindle microtubule attached to the centromere, so now you can see that this structure, the one I highlight now, this structure is known as the chromosome. So the chromosome moves towards the centrosome away from the metaphase plate, and due to the attachment of the spindle microtubule at the kinetochore of the centromere, so you can see that centromere move first. So at this moment, how many DNA molecule per cell? The answer is 92 because there is no DNA replication and there's no separation of the cells or cytoplasm yet. So therefore, the number of DNA per human cells still remain at 92, and the number of chromosomes here remain at 46. So what are the behavior of chromosome during anaphase 1? So homologous chromosome now, they are separated at their chiasmata, breaking the connection formed during crossing over. And each chromosome is pulled to opposite pole of spindle. And remember, it's chromosome, not sister chromatid, because at this moment, this chromosome still made of two sister chromatid joined at the centromere. So chromosome now move away from the metaphase plate towards the centrosome at opposite end of the spindle. And because of the attachment of this spindle microtubule to the centromere, so therefore the centromere always move first and the chromosome are dragged toward the pole because the spindle microtubule attached to the centromere region. So how about centrosome? So centrosome remain at the opposite pole of the spindle throughout the anaphase 1. And how about spindle microtubule? Spindle microtubule is shortened, pulling the chromosome towards the opposite pole of the spindle. As they shorten, the tension causes the chiasmata to break, allowing the homologous chromosome to separate. The nucleolus remain absent during anaphase 1, and the nuclear envelope remain disintegrated throughout anaphase 1, and there's no event associated with or to plasma membrane, and the plasma membrane remain intact during anaphase 1. The next phase is the telophase 1, and during telophase 1, you can see that the nuclear envelope start to reform, nucleolus start to reform as well, and these the chromosome arrive at the opposite end, and they start to uncoil into chromatin in some of the species. So at this moment, cleavage furrow is formed by this the plasma membrane. However, the cell is not divided yet during telophase 1. Normally, cytokinesis takes place at telophase 1, but the completion of the cytokinesis takes place at the end or after the end or completion of telophase 1. So the number of DNA molecule per human cell at this stage is still 92, and the number of chromosome per human cell is 46. However, at the end of telophase 1 and also cytokinesis, this number will be half. So what are the behavior of chromosome? So chromosome arrive at the opposite pole of cell, and this established two distinct sets of chromosome, each with one chromosome from every homologous pair. In some species, two separate nuclei begins to form around each set, and in certain species, chromosome begin to uncoil into the longer, thinner, and less visible chromatin fiber. How about centrosome? So the centrosome remain at opposite pole of the cell, and the spindle microtubule are broken down as their role in pulling chromosome is complete. In some species, nuclei are reformed due to the uncoils of the chromosome to form chromatin. And the nuclear envelope is regenerated around each set of chromosome in some species, forming two haploid nuclei now. Yes, a cleavage furrows form in animal cells, beginning the process of the cytokinesis, where the cytoplasm divide. Next, let us look at cytokinesis and chromosome status following meiosis 1. So cytokinesis, also known as the divisions of the cytoplasm, and it typically occurs at the same time as telophase 1, resulting in the formation of two haploid daughter cells. So in animal cells, a cleavage will form, pinching the cell's membrane to separate the cytoplasm to form two cells. However, in the plant cells, a cell plate develops between the nuclei to form new cell wall, which separates the cytoplasm into two cells. At the end of the meiosis 1, the chromosome number is half. So for humans, 46 chromosome is divided into two cells. Okay, so therefore each cells contain 23 chromosomes or unpaired chromosome. So in humans, this means the chromosome number is reduced from diploid to n equal to 46 to haploid n equal to 23. So although the cells now are haploid, but each chromosome still consists of two sister chromatid joined at the centromere, which means that each cells now still have 46 DNA or 46 chromatids. So therefore, meiosis 2 is needed because these sister chromatids will be separated during meiosis 2, contribute to produce four genetically distinct haploid cells by the end of the meiosis. And with that, we have come to the end of this lesson. Hope you found it helpful and easy to follow. If you have any question, feel free to drop them in the comments section. See you in the next lesson and have a great day.